Welcome everybody. I will now come to a very important topic in network analysis, which is called module preservation analysis. And so, as I mentioned in an earlier lecture, module preservation is often an essential step of a network analysis. You may remember that module preservation is one of the critical steps in this workflow, and where, on the one hand, if you have similar data, you do carry out module preservation in order to check the robustness of your module definition. You just want to tell yourself and your reviewers that, yes, this is a biologically meaningful module. But also, module preservation can be very exciting to study whether things change from one condition to the next. For example, you may want to find a module that is only present in female brains but not in male brains, or female liver and not male liver, or in human brains but not in chimpanzee brains, and so on. And let me come to a motivational example. So here we want to study the preservation of human brain co-expression modules in chimpanzee brains. And the data came from a publication by Mike Oldham. And so Mike Oldham had, as I mentioned, these two different data sets, human brain expression data and chimpanzee brain expression data. And what one can do then is to apply WGCNA to identify these modules in human brains and in chimpanzee brains. And then, of course, the natural question is, well, what's the relationship? And one standard approach for addressing it is to create such a cross-tabulation table where the rows report human modules and the columns report chimpanzee modules. And the entries of the table report how many genes are shared in common. For example, there is the black human module, which originally contained 50 genes. It corresponds to the black chimpanzee module, which by itself contained 37 genes, and the overlap between these two modules is 23 genes. Now, the other thing you can then calculate is some hypergeometric p-value, and it turns out to be a p-value 10 to the minus 31, you know. And so in this way, you can calculate for each pairing of modules what is the overlap. Now, incidentally, you see here that there almost is a diagonal of red, right? Which indicates that there's actually very good agreement as long as you reassign labels in the chimpanzee data so that they match the male modules. Do you get that? So when you do a cluster analysis, then you are certainly free to change the labels of the clusters because they have no intrinsic meaning. And often it greatly enhances the reader friendliness of your scientific paper if you do that analysis so that modules agree with each other, you know. Can you imagine you write a paper about humans and chimpanzee brains, and then the black human module corresponds to the blue chimpanzee module? Think of forming a sentence with that, you know. So therefore, we have an R function which is very useful for matching labels of clustering, and it's called match labels. And it's a trivial function, but it is very useful, you know. And it does what it says. It matches labels according to the overlap based on the hypergeometric test, you know. And I will, of course, illustrate the use of this function tomorrow in the R software tutorial. Yes?
Two questions. Do the modules enrich similarly between the species?
Yes, yes.
And then
yeah.
how come there aren't more gray modules, gray transcripts?
Yes, yes, okay. here, so there's an expert in the audience who noticed there's a gray module here, which, remember, gray indicates genes that are not part of any module. And there are only 39 transcripts in there, which feels low. But the reason is the following, that Mike Oldham is really an expert in pre-processing data, and he decided to build this network using only transcripts that are highly expressed in brain tissue. So he pre-filtered these genes heavily. I don't know the exact number, but I want to say these are about 5,000 genes or so, you know. And yeah, there's your answer, you know. Just looking at the dendrogram, you notice there's barely any gray. Here's the gray, you know, which tells you that most genes actually end up in a module, you know. But yes.
Very briefly, how did you arrive at the same number of modules for each?
We didn't force it. That's what we found.
So if that does not happen, then
no problem. No problem. So that's another question was, notice the number of modules for humans is very similar to that for chimpanzees. I didn't count them, but there's no requirement whatsoever that this is the case. You could have 10 human modules and 30 chimpanzee modules, no worries. I mean, in this case, it would, of course, the match labels would try to align it as good as it can, and the rest remains distinct, you know. Yes?
It looks like it's a static cut.
Yes, that's right.
So then you do
good eye.
specify
Yeah.
modules.
Yeah, so so notice there's this horizontal red line, which indicates where the branches were cut. And this analysis was actually done in 2005, 2006, when we didn't have the dynamic branch cutting method, you know. And so we had to take what we had, you know. But we got away with it because these branches were very distinct. And no harm done, you know. But I completely agree with you. The dynamic branch cutting would have been more appropriate, you know. Yes?
When you're looking at preservation across two different species or different arrays, how are you matching? Are you matching on like a gene level? Or?
Yes, that's a good question. So when we compare the module structures of two data sets, then we need to first agree on what is a node in the network, you know. Now, if you use the same technology, say Affymetrix array, then that's an easy exercise. You just match by probe identifier. But if you have two different technologies, Affy versus Illumina, for example, then what you can do is you can match at the level of on-trace ID. You know, so Jeremy Miller, who is in the audience, he wrote another very valuable R function called collapse rows. And so it allows us to collapse, let's say, 50,000 probes into 21,000 genes, where each gene is represented by one probe. And there's a way to pick it. And then you match data by on-trace ID, you know. But imagine you had two different species, mouse brains versus human brains, then you would need to find orthologous genes and match that, you know. So that is actually, one carefully wants to think about how to match it. But everything I describe assumes you went through this painful way of matching things. But I do refer you to the R function collapse rows, which is in the WGCNA R package, and there's also an article described about it, you know. Other questions at this point? Or, yeah. Here for the human chimp analysis, Mike Oldham used data where the authors had filtered the probes so that they had sequence equality, and they could be found in both species, you know. It was a very careful matching. All right, but let's now think of our challenge of writing a scientific article, you know. So let's say, so we find this, and what do we see? Most modules really correspond to each other. But let's say there is this one module, the red module. Do you see? It seems to split into two modules. So the human red module contains 122 genes, and it splits into the red chimpanzee and the yellow chimpanzee module. And if we were bold, we would say, wow, there's this module that seems to do something different in chimpanzees. But let's imagine we had now found a module that is actually only present in humans, but couldn't be found in the chimpanzees at all. In principle, that could be a very exciting module. But what would the reviewers say when we submit our manuscript? They would say, well, the reason why you didn't find this human module in chimpanzee brains is because you used the wrong branch cutting algorithm. Here you use the static cut. If you had used the dynamic hybrid cut, you would have found the module, Or they would say, you shouldn't have chosen a power of 12 if only you had chosen a power of 8, So it would be very difficult to argue to a reviewer that the module is not preserved, And so these standard cross-tabulation-based statistics have these severe advantages that, on the one hand, you can't make strong statements of non-preservation, and also, relatedly, they're only applicable when modules are defined via a clustering procedure, And so, therefore, we argue that network-based statistics are superior for studying module preservation. It's actually a major reason why weighted networks tools greatly enhance cluster analysis, And so now I describe results from this article whose title says it all. So, is my network module preserved and reproducible? And in this article, we took a very broad definition of a module. We have a very abstract definition, which is a module is simply a set of nodes, any set of nodes, just a set of nodes in a network. For example, a module could be a KEGG pathway, apoptosis genes, or it could be a gene ontology category. But of course, another definition of a module could be a cluster, So module is any set of genes. And then to introduce some terminology, these module preservation statistics are then used to assess whether a module that you found in your reference network is also preserved in your second data set, the test network. So for example, if we wanted to focus on human brain modules, we would choose those as the reference network and ask which of them can also be found in chimpanzees. And that would be the test network. Another example comes from mouse genetics. So these are again data from Jake Lucis's lab, where Jake generated an F2 mouse cross and collected multiple tissues, so liver tissues, adipose tissue, muscle tissue, and brain tissue. And here we defined the module as the set of cholesterol biosynthesis genes, And so these labels here are known cholesterol biosynthesis genes, but the edges correspond to co-expression patterns. So we used, for example, here female liver tissue, and a red line indicates that these two genes are highly correlated, positively correlated in female livers, and a green line indicates that the two genes have a strong negative correlation. And let's assume we take the female liver as, so this network in the middle, as the reference set, and we ask, well, how similar does it look to any of the other networks, And I hope you notice that the female liver network looks most similar to the male liver network. No surprise here, And but its relationship to brain tissue is very different, And what we do here visually is really what we call module preservation analysis. We say, well, the connectivity pattern is highly preserved in male liver, but not preserved in female brains or male brains, you know. And so I will then, in the following, describe to you these preservation statistics that we use for assessing it, you know. So in order to measure relationships between different networks, for example, the female liver network and to the male liver network, we use what we call either network concepts or network statistics. And one network concept is already very familiar to you, which is just the connectivity, the number of friends that a gene has in the network. Another measure of another network statistics is called density. So what is that? The density is just the average adjacency. So in a social network, if you have a clique corresponding to a module, the density is just how much do these people like each other. So that if you take the high school club, they all, the science students, they hopefully love each other in high density. But then there's another module, the political debating society, and they split into various camps of great acrimony, and their density would be lower, you know. Another network statistic, using these network statistics, then you can assess relationships between networks. For example, when you have this connectivity in the reference network and the connectivity in the second test network, you can simply correlate them, you know. And what does that measure? It measures, among other things, preservation of hub gene status, right? If a gene is highly connected in the reference network and then remains highly connected in the test network, then the hub gene status is preserved. You can also assess density preservation. So let's say you have a module that is highly or densely connected in the human brain. You can ask, do these genes remain densely connected in the chimpanzee brain? Other things you can do is you can literally take the adjacency matrix in the reference data and turn this matrix into a vector, do the same thing in the test network, and just correlate these adjacencies matrices with each other. So that measures connectivity preservation again, you know. So our module preservation statistics can really be applied to different types of networks. For one thing, you can certainly apply them to protein-protein interaction networks. You can apply them to unweighted networks. However, particularly powerful statistics are available for correlation networks, as I will show in the following. And so these weighted correlation networks are particularly useful here because you want to detect subtle changes in connectivity patterns, you know. So these network-based module preservation statistics take really three inputs. The first is the module assignment in the reference data set, right? You start with the human brain modules. The second input is an adjacency matrix. You need to know the connectivity pattern in the reference network. And then you need an adjacency matrix from the test network. Why? Because you want to study whether the connectivity pattern is preserved. But the one thing to notice is what it doesn't require. It doesn't require the module assignment in the test data. Do you see that? Nowhere do I have to specify what are the modules in the chimpanzee brains. I just need to say what are the modules in the human brains, you know. And so by not requiring us to specify the module assignment in the test data, we free ourselves up from the vagaries of branch cutting and all of that. And we can rigorously say it doesn't matter how you cluster things in the second data set. Our statistic indicates the module is not preserved, so it doesn't matter, you know. Yes?
If I flip the two data sets,
yes.
Suppose I use the test data set as a reference.
Yes.
Not change.
Yes. So the question is, what if you flip the role of reference and test data, you know? And it has a big effect, you know. Let's stick with our example of the human-chimp comparison. Of course, in Mike Oldham's paper, he really focused on human modules and then studied their preservation in chimps. But somebody else may have said this is too human-centric. I carefully annotate modules in chimpanzee brains, and if they ran the analysis, it could turn out that they find a module that is not preserved in human brain data, and that could be a very exciting finding, you know. And I do know of a doctoral student who wrote 2 papers, you know, one where he took the one reference set and then reversed it. And we laugh because it feels like you get 2 papers out of one data set. But the truth is, when you write these papers, they are actually lengthy papers, you know. Why? Because you get 10 modules, you need to explain to a reviewer what's the meaning of the modules, you need to validate things. You very quickly end up with 20 pages. And then if you reverse it, you have another 20 pages, you know. So if you want to be kind to your readers, you actually split it into 2 papers, you know. Other questions? All right. So what do these network preservation statistics measure? They measure really 3 broad categories of properties. The first is, as I mentioned, network density. So does my module remain densely connected in the second data set? The second category is connectivity preservation. So is my hub gene status preserved, for example? But the third category I didn't mention yet is to study the separability of modules. So let's say you have the human brain data, and you find that these 2 modules that 2 modules are very distinct. You know, they're separate. But then when you and you find that both modules are also present in the chimpanzee brain. But it could be that suddenly they are no longer distinct. They seem to merge, you know. And so we also provide separability statistics, where you could say to a reviewer, if you want to make the argument, no, my module really remains distinct. And earlier today, Peter Langfelder gave a talk about what he called consensus network analysis and eigengene networks. These eigengene networks are all about point number 3, you know. 2 modules that are separate in one condition, do they remain separate in another, or do they merge, or do their relationships change? That's when you use these eigengene networks and preservation statistics, you know. Now, in my talk here, I actually don't care about separability of modules. We often ignore it because the first 2 questions turn out to be often far more important. Why? Because you have a super exciting module in one condition and you just want to know, is it present in another? Never mind whether it's how it relates to other modules. All right, so what kind of connectivity preservation statistics do we have? You can form seemingly endless ones. Remember, we have this measure of intramodular connectivity, and so you could just correlate K intramodular in the reference data with Km in the test data. And so is intramodular hub gene status preserved? As I mentioned, you can correlate the adjacency matrices. Now, if you have a correlation network based on an underlying correlation structure, and you can form the correlations of correlations, right? So you would vectorize these matrices and then just correlate them. And also remember, we have this measure of module eigengene-based connectivity, the KME measure, and of course you can also correlate this measure from one condition to the next. So by now, probably your head is spinning, so many statistics. But it turns out that one can derive relationships among those in case of correlation networks. They roughly measure the same thing, you know. And so therefore we aggregate them, you know. Instead of writing a paper where we output 4 statistics, we aggregate it into one overall statistic, and we're getting there, you know. One thing to notice is all of these statistics are defined so that the maximum value is 1. Why? These are all correlations, and 1 means perfect preservation, you know. So we actually have 4 connectivity preservation statistics, 4 density statistics, and all of them take on values less than or equal to 1. And what we really need are thresholds, you know. We want to have a significant threshold. Why? Because we want to make a statement this module is significantly preserved or not. And to arrive at significant thresholds, we use an old trick, which is a permutation test analysis. And it's a very trivial idea. So if you have the gene labels in the test data, meaning, for example, the chimpanzee brains, you just randomly permute these gene labels in the chimpanzee data, and you calculate the module preservation statistics. And what does that accomplish? Well, this is the value of the module preservation statistic under the null hypothesis, you know. And so you repeat that exercise 200 times, and so you can then calculate a mean value of your module preservation statistic. Similarly, you can calculate a standard deviation. And so what we then do is we calculate a z-statistic for our module preservation statistic. So we take the observed value minus the mean divided by the standard deviation. So what does that measure? It measures how far away is your observed value from the value under the null hypothesis. So remember, we have multiple statistics, let's say 8 different preservation statistics, and so we get 8 different z-statistics. And each z-score provides an answer to the question, is the module significantly better than a random sample of genes? And, but we don't want to report 8 statistics. And so we summarize these 8 statistics into one overall statistic called z-summary. And for that statistic, we have thresholds. And we say, well, if z-summary is less than 2, then there is no evidence that the module is preserved. And that's a wonderful statement to make, because we can say no matter how you cluster your data or what transformation you apply, this module is simply not preserved. The connectivity pattern is not preserved, the density is not preserved. In contrast, if your z-summary statistic value is bigger than 10, we say there's strong evidence that it's largely preserved. You know. So let me come, I skip a few slides, and I just want to give you some details for one particular statistic, because there are some people here who are very good at math, and so I just wanted to give you that as an insight. So let's say we have this one preservation statistic called core.core. Remember that's the correlation of correlations. And Q denotes the Qth module. So now core.core is an observed value, and let it be 0.9, highly preserved. Now, as I said, we calculate the expected value of core.core under the null hypothesis using this permutation test. And similarly, we get the variance under the null hypothesis using the permutation test. So that's z core.core. Now, analogously, we have these other connectivity statistics, core.kme, core.adjacency, core.intramodular connectivity. But as I said to you, they all measure the same thing. So you might as well form just an aggregate measure of it, and that's the median. Do you see? So we end up with a statistic called Z-connectivity that aggregates 4 different statistics that all measure connectivity preservation. Similarly, we have 4 statistics that measure density in a module. The mean correlation, the mean adjacency, proportion of variance explained by the module eigengene, mean KME. And again, we form the median, and so we get Z density. And then, in order to get Z summary, we form the average of Z connectivity plus Z density. Do you see that? So the Z summary statistic really assesses 2 properties. It assesses is the connectivity pattern preserved and is the density preserved, you know. Any questions or comments? Yeah, I just want to tell you, this will become very biological soon. So for the biologists here, don't run out. And I should tell Peter to close the door, you know. But there's some people that do like math formulas, believe it or not. So in any event, so that's the Z summary statistic. The Z summary statistic, to tell you already, has one property that may be unattractive. And that the property is that it depends on the module size. And let's think about it. Imagine you have a module that contains 3 genes. And then you find the correlation patterns between these 3 genes as preserved from humans to chimps. Will you be impressed? No, because you say, come on, just by chance, of course this will happen, you know. In contrast, if we have a module comprised of 300 genes and we find the correlation pattern is preserved, yes, we will be very impressed, you know. So, so clearly module size matters when you judge statistical significance. And therefore, yes, the Z summary statistic will depend on module size. Now, although this is a feature and not a bug, it's sometimes unattractive. And why is that? Because in some applications, you have a relatively small module of interest. You may study a very complex disease, schizophrenia, and you find a module that contains as few as 30 genes. But you still want to assess module preservation. And so what you need is a statistic that really is independent of module size. And here it is. This statistic is called the median rank statistic. It's really defined in an analogous fashion as the Z summary statistic. However, it does not require a permutation test. And how is it defined? It's actually very intuitive. Remember, you have 8 different preservation statistics. And so let's say you have 20 modules. You can rank each module according to these 8 statistics. You can say this is the most preserved module, this is the least preserved module. And so you end up with 8 different rankings. And then you form, in some shape or form, medians. You get the median ranking, you know. The median defined in this analogous fashion as I have here. You form a median here, and average it. And that statistic, the median rank statistic, shows no dependence on module size. And it is, of course, calculated in a very fast fashion. Why? Because you don't have a permutation test. You just rank things. However, the drawback of this statistic is that you can never argue that a module is not preserved. You can say this is the least preserved module, but it could still be preserved, you know. Or you can say this is the most highly preserved module, but it could be that none of the modules are preserved, you know, because the median rank statistic doesn't have a significant threshold, you know. So what I just said is summarized here. What you need to take away is there are 2 composite statistics, Z summary and median rank. And although this talk may have been slightly complicated, I give you R code that accomplishes it. And you need to become an intelligent user of reading that output, you know. So let's come to an application. Now we become a little bit more biological. So coming back to the comparing human brain data and the chimpanzee brain data, but now I define modules as KEGG pathways. And why do I do it? I want to show you that we don't need modules defined as clusters. You can have KEGG pathways. However, the connectivity pattern between these genes is given as a signed correlation network. And this is a typical output of such an analysis. So the y-axis shows the module preservation statistic, Z summary, and the x-axis shows the module size. And each dot corresponds to a different KEGG pathway. There's, what do we find? Well, there's MAP kinase, KEGG pathway, calcium processing, Wnt signaling, and so on. And all of them have a Z summary statistic around 6 or so, meaning moderate preservation. You know. Actually, the module sizes are rather small, and so this is pretty high preservation. Now, the one dot that is not preserved, do you notice there's this dashed line shows the significance threshold, and notice apoptosis genes are not preserved between humans and chimps. That's interesting, you know. Now, the apoptosis module here contains very few genes. I want to say like 23 genes. And you could say, well, maybe this is an artifact of that I use the Z summary statistic. So therefore, we now go to the median rank statistic. And what we find is that the apoptosis module was the least preserved module. It has the highest rank, you know. For people with very good eyesight, I want to draw your attention to the y-axis of the median rank statistic, because Peter Langfelder did a horrible crime here, which I see the y-axis values typically go up, right? And whereas here the rankings, the rank 8 is down, you know, which shows that Peter is a phenomenal R programmer, you know, to accomplish it. But personally, when you read my software tutorials and you use the median rank statistic, then the things at the top are always the least preserved ones. But the reason why Peter arranged it in this fashion, because he wanted to highlight that the apoptosis is at the bottom of the barrel when it comes to preservation, you know. Just be aware of it. So in any event, we found apoptosis seems to be different. Now, never trust statistics. Always visualize everything, right? So here, Peter visualizes the connectivity pattern between the apoptosis genes in the human brain data, and similarly, on the right-hand side, the connectivity pattern in the chimpanzee brain data. And I mean, just looking at it, do you feel that this is preserved, you know? Not really. I don't see anything is preserved, you know? And so, which tells us that, yes, our statistics work. They detect there's no preservation. And at this point, of course, you would want to somehow validate it. You would want to validate that, yes, apoptosis genes in human brains have a different pattern than from those in chimpanzee brains. And if you have a big lab, you can do that. But I just had Google, you know, so I did a Google search, which is Neuron apoptosis human chimps, you know. And I dug out some interesting references. So there is an article that asked the question, did natural selection for increased cognitive ability in humans lead to an elevated risk of cancer? So what's going on here? We as human beings, we really need our neurons. We can't kill them off. And so therefore we want to lower apoptosis. They should stay alive. But that comes at a cost, because as you know, apoptosis is a major mechanism to fight cancerous cells. You know, if a cell looks funny, one triggers, hopefully, the apoptosis pathway. And it seems then in chimpanzees, they have a more active apoptosis mechanism for example, chimpanzee tumors are extremely rare and biologically different from human cancers. Chimpanzees are actually very bad model organisms for studying human cancer, you know. And also, there were publications that showed that apoptosis genes show strong evidence for positive selection. So there is really a difference between humans and chimpanzees when it comes to apoptosis. And so this then suggests to me that our findings could be real, you know. But the other thing I learned from this exercise is there is a journal called Medical Hypotheses, you know. So, so if you have a great hypothesis, go ahead and submit it there, you know. So, all right. So let me now come to this other example that we started earlier. Remember this plot, the cholesterol biosynthesis genes in different mouse tissues. And let's just check whether the female liver module is most preserved in the male liver. And this is reported here. So here on the panel A shows on the y-axis the z-summary statistic, and the x-axis shows different tissue gender combinations. And what do we see? Well, we see that the female liver is most highly preserved in the male liver. Z-summary statistic close to 10, you know. Whereas there's less preservation in other combinations. But now, using these statistics, we can ask a more subtle question. We can ask, well, what is the density preserved? And so what we see from the panel B is that the density is high, of the female liver module is highly preserved in the male liver, but not anywhere else, right? But interestingly, something is going on with the Z connectivity statistic in panel C. Notice when you look at hub gene status preservation, connectivity preservation, actually there's some moderate evidence that is also preserved in adipose tissue. Do you see that? And so, do you see, these statistics allow you to address question, well, what is preserved or what is not preserved, you know? And so there were some papers where we found a very exciting module, and we found that the density was highly preserved in other data sets, but not connectivity, you know, and vice versa. So it's actually a good idea to separate these. But not to confuse you, the workhorse of module preservation analysis is the Z-summary statistic. You know, if you want to be carry out a straightforward analysis, focus on that. So now I give you another biological application carried out by Jeremy Miller and Dan Geschwind. And as you know, Jeremy Miller is actually one of the instructors of this course, so he will give you several talks tomorrow. And so in any event, here Jeremy and Dan compared human and mouse brain data. And so Jeremy collected all the human brain data he could in Geo, and similarly all the mouse brain data that he could, and he formed then two networks. One is the human brain network and one is the mouse brain network. And he wanted to assess, well, which human brain modules are preserved in the mouse brain and so on. Now, why would we do that? Well, because modules often correspond to cell types, and then looking at hub genes that are present in preserved modules could lead to reliable markers for cell types across species. But also, Jeremy wanted to understand why certain diseases are enriched in human populations. For example, Alzheimer's disease is unfortunately a disease that afflicts humans, but not mice, you know. So Jeremy then formed two plots. One is to study the relationship between the mean expression in mouse brains versus mean expression of a gene in human brains. So each dot is a gene, and you just ask, is the mean abundance preserved? And he found a correlation of 0.60, which, if you're an aficionado for these comparisons, that is a very high preservation, you know. I wouldn't have expected it. In any event, but it reflects one thing, that Jeremy was very careful with cleaning the data and pre-processing it. You know, it required substantial work to get to reveal actually a fairly high preservation of the mean expression level. But then you could ask another question, which is the connectivity preserved? Right. So here the right panel shows on the x and y axis the connectivity of each gene in the human and mouse brain data. And what do we find? Well, much less preservation. There are some genes that are highly connected hubs in human brains, but not in mice, and vice versa. And when you find that connectivity is not preserved, it always means one thing, which is module structure is not preserved. There's some modules that are not present in the other condition. And so Jeremy did then this more careful analysis where the upper panel shows the modules in the human brains, and the lower panel shows the modules in the mouse brain. And again, if you carefully look, Jeremy used the match labels function to assign the same colors to these modules. You know, for example, this human module 15, which is yellow, corresponds to the mouse module 15, which is also yellow. That's not an accident. This was the colors were matched why to enhance reader friendliness. You know. And but in any event, he found two modules, module 7 and module 9, which are these small slivers here, that were very exciting because they were enriched with known Alzheimer's disease genes. And they could be found in the human brains. However, these two modules could not be found in the mouse brains, you know. And so in principle, we could now write a paper, submit it, and say, wow, we found these two exciting modules in human brains, and they are not preserved in mouse brains. But then the reviewer will carefully look at it and say, oh, come on, this is this tiny module here, you know, and you don't find it here. Well, do a different branch cutting algorithm or something, you know. So it was really critical to use these module preservation statistics to rigorously argue that no matter what you do in the mouse data, you will not find these modules. You know. And so in his paper, Jeremy shows then here these different human modules, and this column reports the Z summary statistic. And look, this module 7 and module 9 could not be found because the Z summary is 0.3 and 0.4, well below the thresholds. So we could rigorously argue these modules are not preserved in mice. However, just to mention it, these modules could be found in multiple human data sets. So they exist without a shadow of a doubt. You know, these are real modules in humans. All right. Now, one thing you could think is the following. Well, the WGCNA, it clearly has value in heterogeneous tissues. Why? Because we find these modules in brain tissue, which, for example, let's just look of how Jeremy characterized them. There's some modules correspond to oligodendrocytes, some astrocytes. There's a module microglia, there's a module of neurons, another module corresponds to ribosomes, and so on. So they're, or mitochondria. So modules correspond to cell types, to cell compartments, and so on. But you could say, well, what about single cell analysis? If you literally have the mRNA from a single cell and you apply WGCNA, what do you find? And fortunately, I have a colleague here Guoping Fan and a student Kevin Huang and his collaborators from Beijing University. And they had a very exciting data set, which is single-cell RNA-seq analysis from pre-implantation embryos, okay, in humans and in mice. So let me give you a little bit of a background. So mammalian pre-implantation development is a highly complex process. So just to remind some of you, or some of you have never heard of that, but you start with a fertilized egg, an oocyte, and then there comes different stages: pronuclear, zygote, 2-cell, 4-stage, 8-cell, morula, right? And then it becomes an embryo. And so Guoping Fan and his lab, they had human data from single-cell data from a fertility clinic, in essence, and these are the sample sizes from different stages, and this is a principal component analysis. So what do we see? There were 3 single-cell samples from the oocyte stage, and then several from the pronuclear and zygote stage. And when you do this PCA analysis, it's interesting that these samples line up according to stages, you know. But we really don't learn much from this PCA plot, because it just tells us what we know from our textbook, that yes, that first it's an oocyte and then eventually morula. What we want to know is, well, what are the modules, what are the big themes in the transcript of these data? And so here we applied WGCNA then to this data set. And the other thing I think I forgot to highlight, these are RNA-seq data, because people sometimes ask me, are these methods valuable for RNA-seq data? And so it's a resounding yes, you know. I have several examples where they work very well. But in any event, so what do you see? Look at this hierarchical cluster tree. We see very distinct modules, very distinct branches, you know, which tells us in this signed network analysis, there's a clear structure. Now, the next thing one can do is one can visualize then these different modules in terms of their expression across different stages. So in contrast to our usual convention, the genes in this heat map are in the columns, and the different samples are rows. And what do we see? There's this one module which had the color BISC4, you know, which I don't know what BISC4 looks like, but that was the color. And no, this is what BISC4 looks like. There's actually, here's the color at the bottom, in case you care to know. And so what do we see? The BISC4 module contains genes that are highly expressed in the oocyte stage, right? You see a lot of red, and then it tapers off. It goes down to low expression. Other modules are clear. So for example, here there's the pale violet 3 module contains genes that are highly overexpressed in the morula state, you know. And so what do we learn? Largely, many of these modules really contain transcripts that are highly upregulated at particular pre-implantation stages, which is perhaps expected, but it's interesting, because we now have stage-specific transcripts, and we can look at transcription factor enrichment or transcription factor binding sites to understand, well, what upregulates these genes. At the very simplest level, we can look at hub genes inside each of these modules. Also, of course, you can see how these module eigengenes differ across these stages. And so here, the module eigengene is based on the RPKM values. And so this the oocyte module shows this pattern. And as expected, the module eigengene takes a high value in the oocyte stage, but then lower values elsewhere. Here is another module, the 8-cell module, and yes, all the transcripts are highly abundant at the 8-cell stage. All right, now the reviewers of this article wanted us to show that these modules are preserved, fair enough. And so fortunately, there were public data sets out there, microarray data, and you can just ask, well, are my RNA-seq-based modules preserved in these other types of data sets? And so we calculate here the Z-summary preservation statistics in the left panel. And so what do we find? Well, these stage-specific modules that we care about a lot, they show very high preservation in other human data sets, which correspond to different columns. But interestingly, some of them are also highly preserved in mouse data sets. And that was one of the take-home messages of this paper, that stage-specific modules in humans show high preservation in mice. So that's the Z-summary statistic approach. And but you can also do a cross-tabulation-based analysis. So we have R-code. Remember, you've seen that before. So here are the different modules in humans, in mice, and here the columns correspond to modules in humans. And the right panel shows the number of transcripts that are overlapping. So as I mentioned, all of these tools are implemented in the WGCNA-R package. And later I give you a software tutorial for how to carry out module preservation analysis. But I mentioned already there's an R function called module preservation that really does that analysis. And here I focused on the Z-summary and median rank statistic. But I do want to tell you that this function implements, I want to say, 25 different statistics that you can use to address all sorts of questions, because I, of course, mentioned that we like to free ourselves up from having the cluster, the module assignment in the test data. But the module preservation R function also implements several statistics for assessing module preservation based on 2 cluster assignments. So if you happen to have the cluster assignment from the human brains and the cluster assignment from the chimpanzee brains, you can input that into the function, and it will output a variety of cross-tabulation-based statistics. Let me ask here, do you have any questions at this point about module preservation? Okay. Are you all traumatized? You will appreciate these methods when you analyze a data set, because I knew students who spent their dissertation project trying to argue that some modules from one tissue are not preserved in another tissue. That was a 3-year project, painful. And but nowadays we have an R function. That's a what? That's a 2-hour analysis, and it's 2 hours because you do a permutation test. So so in this sense, it's valuable. I do want to also mention that the module preservation statistics are very good for assessing whether modules from one condition are preserved in another condition. However, sometimes that's not what you're really after, but you rather want to know, to have modules that are present in multiple conditions. Peter mentioned earlier an application 8 lung cancer data sets, and you really don't care which modules could be found only in AFI data but not in Agilent data, right? That really doesn't interest you. No, what you want to know, give me the modules that are really present across all studies. And if you have that goal, you don't do module preservation analysis. That's the wrong technique. In this case, you really want to do consensus module analysis, So keep that in mind. These two tools address different research aims. All right. Let me. I'm not sure how I am on time. I would like to take another 5. Am I fine? Okay. I want to mention another topic, which will become interesting when we have real data in our R software tutorial. And so, as you know, we kept talking about gene modules, meaning modules of genes, and which implies that we construct a network among genes. But some of you may have thought, well, why don't we flip things around? Why don't we transpose the data and instead construct a network among people, right? It's just a transpose. And it's an obvious idea. And so Mike Oldham explored the utility of that idea. And so we describe these approaches as sample networks. Why? Because the nodes correspond to samples, human subjects, for example. And so remember, we earlier discussed the Huntington disease data set from Hodges et al., which is actually a very complex data set. And why is that? Well, there were multiple brain regions, and also the data came from different countries. There were different batches. The data were processed in different batches. There were the usual differences in age, sex, and also different quality scores. Complex data set. And so we wanted to really process the data using a network approach. And so what we can then do is we can construct a sample network as a signed network. But now look carefully. What has changed is that here the entries are samples. So these are vectors that may have 30,000 entries. Why? Because for one person, these report 30,000 gene expression levels. And now, when you construct a sample network, you want to choose a low threshold. Remember, the default threshold for a signed network is 12. And why is that? Well, because gene networks contain thousands of genes, and you need a threshold to emphasize, to protect against false positives. But when you're dealing with samples, you may have, 50 samples. The rationale for choosing a threshold is not as compelling, and there are reasons to choose a low threshold. So we choose here essentially a power of 2. And then we can, of course, again define the connectivity of a sample. And what's the interpretation? Well, if a sample has a high connectivity, then it means that it looks very similar to all the other samples, meaning probably this is a high quality measurement. In contrast, if you have a sample that is not connected to any other sample, low connectivity, maybe it's an outlier, or maybe the RNA integrity was poor, or there are batch effects, something went wrong. And so in order to arrive at a measure of outlyingness, what we do is we take the connectivity and subtract the mean from it and divide it by the standard deviation. And that measure then is, so what does that measure? This is what we call, again, a Z statistic, right? You take, you standardize it. And when you have a negative value of Z dot K, then it means the sample is outlying. So that's a sample network statistic. And so when we, here we plot then the Z dot K for different samples. And what we see is there are many samples that appear to be outlying because their Z dot K value is less than -3 or close to -4. So they appear to be outlying until you look at what these samples are. So these samples are caudate nucleus sample from affecteds. And remember, Huntington's disease affects the caudate nucleus. These neurons die off. And so actually there's a very good biological reason why these samples are outlying. Well, the disease makes them outlying. And therefore, we would not remove them from the analysis, you know. In this outlying here that's the signal. If you removed those, you would have removed the signal in your data, you know. Here I acknowledge my many collaborators. I'm glad several of them will present here. One is, of course, Peter, who presented some of the material, and also Jeremy Miller will present later on. And with that, I would say let's take a little break and resume in maybe 10-15 minutes. Okay, thank you. Thank you.